Siegel, Chair of Friends of Cancer Research. Welcome. This is a big and important meeting, and we're very excited about it. It's hard to believe that this is our 11th year. And just a historical fact, when we started doing this meeting, we were convening on important issues. And it was someone from NCI that we asked to speak, and he said, I'll go, but I'm so bored about speaking at meetings and nothing ever happening. And we thought a lot about it, and we changed our format to really reflect consensus and the ability for people to work together to come to this meeting where, in fact, we could have ideas that were thought out and at least have an open forum, and that was enormously helpful. It also cost a lot of work, but it was really worth it, and we had real results from these meetings. I want to thank our supporters, ACR and ASCO, for your support. Thank you very much. We have white papers that we've all had and you have today on the really important subjects, and I think I'm not going to take you through memory lane, but there's been a huge amount of outcomes from all of these meetings, but none of it would have been possible without your support, your help, without the extraordinary leadership and staff of Friends of Cancer Research, Jeff Allen, Mark, Ryan, everyone in our team has done an enormous amount of work. I mean, if I start to name names, I'm going to forget a few people, and then they'll be mad at me. But mostly, it doesn't happen without a lot of work and commitment. It also doesn't happen without people working with us and getting their ideas and getting their feedback and really making sure that we have all stakeholders' views, the most important being the patient, because everything we do is about the patient and for the patient. So this is really important. Today's meeting, and I'm just going to go through, the topics. Establishing the role of circulating tumor cells, DNA in cancer drug development, augmenting randomized confirmatory trials for breakthrough therapies with historical clinical trials data, and real-time oncology review. Big subjects, important subjects with enormous outcomes. And again, thanks for all those who have volunteered their time to work with us and who have produced these white papers. The intent of these meetings is to review, to get consensus impact, and then to do something with it, not to just put a paper on a shelf. And often that means legislation, often it means guidances, often it means clinical trials. But the idea is not to do this so there isn't a step forward, and these are complicated issues that are important to all patients and we care deeply. So next, it is my great honor to introduce Anna Abrams, who is I'm sorry. I thought I was introducing Anna first, so then, but they said no, I didn't introduce the subject. Anyway, she is the Deputy Commissioner of Policy Planning and Legislation and Analysis of the FDA. Anna has a very strong background in health on Capitol Hill, in the White House. This is something that she's worked on for years. She knows the subjects backwards and forwards. They are doing extraordinary things. Anna, your work at FDA now is just breathtaking. I don't know how any of you ever sleep, but it's 24/7. The guidances that have come out are just extraordinary. The commitment to public health and to patients and the work is just, again, breathtaking, and more importantly, important to patients, and the ability to explain why you're doing it is really important. So we are thrilled that you're here to give the keynote. I do want to mention that two things. That Anna, we work with her very closely with Breakthrough, and that was not. Everyone loves it now, but it wasn't that easy in the early days. Many people said, well, why are you doing it? Why should you do it? And even companies were saying, what is this all about? But it turned out to be incredibly important, and I don't think any of us even realized at the time how important it was. But Anna, was your hard work when you worked for Senator Burr. Also, 21st Century Cures. That was something that you worked on, and I think we have all benefited from that extraordinary work and what it means to patients and to this community. And everyone here has the same goal, and that's to make life better for patients. They're the North Star, but we don't do it without consensus, we don't do it without working hard, and we never do it without a patient at the table. So Anna, thank you very much, and we look forward to your remarks.
Thank you, Ellen, for that very, very warm introduction, and it is my distinct honor and privilege to bring warm greetings from our Commissioner, Dr. Scott Gottlieb. And if I pause a couple times to cough, I apologize, because even deputy commissioners at FDA get head colds, so thank you for your patience. FDA has long enjoyed a strong collaboration with this organization. Thanks to your leadership, an important expedited development program was added to FDA's toolkit, the Breakthrough Therapy designation, an idea that Friends of Cancer Research was instrumental in realizing, working with partners in advocacy, regulation, drug development, and bipartisan champions in Congress to advance this innovative concept, first as a scientific white paper, and then through the congressional process to become federal law. Today, the program boasts more than 250 designations and over 100 approvals. And about half of those designations are for cancer treatments. I'm proud to have been a part of advancing this tool for patients and the FDA when I worked on this authorizing legislation during my tenure with the Senate Health, Education, Labor, and Pensions Committee. Friends of Cancer Research was also actively engaged in developing parts of the 21st Century Cures Act, specifically intended to consider what more might be needed to help accelerate medical product development and bring new, safe, and effective innovations and advances to patients who need them more efficiently. Of note, Cures authorized the FDA's first Intercenter Institute that focuses on a specific disease rather than a product type, the Oncology Center of Excellence, or what I like to call our catalyst. Breaking down institutional silos in this way to leverage the combined skills of scientists and reviewers across the medical product centers has already yielded results for patients, as you will hear from my colleague, Dr. Rick Pastor, later on today. This is truly an exciting time for cancer research, as we see with so many discoveries being translated into important therapies. Oncology is leading the precision medicine revolution as we move from a field based on tissue of origin towards one based on molecular aberrations such as ALK rearrangements, EGFR mutations, and more recently, microsatellite instability or mismatch repair deficiency. That is a change that occurs in the DNA of certain cells in which the number of repeats of short repeated sequences of DNA are different than the number of repeats that was in the DNA when it was inherited. The many important cancer approvals last year and this year are ample testament to the high level of innovation we're experiencing. In just a short period of time, we've seen both the first and second gene therapy products for cancer and the first tissue-agnostic cancer treatment for those tumors that have certain biomarkers, irrespective of where in the body the tumor originates. And so far this year, we've approved 12 novel oncology therapies, including the 6th immune checkpoint inhibitors targeting the PD-1, PD-L1 pathway, a blockade of anti-tumor immune responses that help the immune system recognize and attack cancer cells. If you drill down and look at the list of these approvals, you see the agency's regulatory efforts to be modern, flexible, and able to respond to changing needs and circumstances, while very importantly still fulfilling our public health mandate to ensure safe, effective, and high-quality products for patients. To harness these opportunities in science and technology, the FDA must create structures and processes that are able to keep pace with medical innovation. And to keep pace requires that we build a dynamic regulatory environment across the continuum of medical product development, from the pre-market to the most post-market phase. In today's speech, I'm going to discuss what the FDA is doing at different points along this continuum to advance policies that will make medical product development more modern, more scientifically rigorous, and more efficient. Consider that tissue-agnostic approval, which stemmed from the fact that tumors with a high microsatellite instability are more likely to respond to immune checkpoint inhibitors, rather than requiring a separate development program for each disease site, which may have taken many years and would likely never have happened at all for some of the more rare tumors. We instead created a single therapeutic approach based on solid scientific understanding of the underlying biology of microsatellite instability. Or look at the ample use of novel endpoints in our recent approvals. Rather than overall survival, we accept progression-free survival, cytogenic response or pathologic complete response. And in one approval this year, we used metastasis-free survival for the first time as an endpoint. Now, given the seriousness of cancer, we've long accepted data from studies that don't always conform to a randomized, controlled, double-blind clinical trial testing the product to placebo and an endpoint of overall survival death, which can take a decade or more to collect. So, it's become more commonplace for oncology studies to compare products to the standard of care, to submit open-label studies, and to allow non-inferiority comparisons. And this year, we saw an approval based in part on data collected from patients who were enrolled in an expanded access program. When your organization advanced to the breakthrough therapy concept, you quite rightly surmised that FDA doesn't simply review product submissions, but can play an important role in helping to guide sponsors long before the submission itself. As a result, companies with cutting-edge therapies which qualify for the breakthrough designation are now receiving much earlier interaction and advice from FDA staff. But FDA can help enhance discovery and translation at even earlier stages of medical product development, as you can see with some of our recent work in oncology to reform the clinical trial process. The traditional drug development paradigm calls for 3 clinical trial phases. A small phase 1 tests safety and dosing. Phase 2 tests short-term side effects and effectiveness over a range of doses. And the large phase 3 assesses product efficacy and monitoring of adverse events. However, clinical trials are becoming more costly and complex to administer, which can make drug development more risky, uncertain, and time-consuming. Overly complex trials can also deter enrollment and delay completion. Modern advances to designing and conducting clinical trials can address some of these challenges. Today, the FDA is working across its medical product centers to facilitate innovative designs and patient-centered endpoints for drugs, biologics, and medical devices. This includes the use of master clinical trial protocols such as basket, umbrella, and platform trials, which involve a common clinical trial infrastructure to study one or more interventions in multiple diseases, or a single disease with multiple interventions, each targeting a biomarker-defined population or disease subtype. For example, I-SPY 2 compares up to 12 experimental therapies in subgroups of patients with metastatic breast cancer with 10 distinct biomarker signatures. This hub-and-spoke design uses a common control, reducing the overall number of patients to be recruited and enrolled. Seamless trials used in oncology are another novel approach. They compress the trial phases into one large, continuous trial. Since a lot of time and cost is spent between the starting and stopping of the traditional 3 phases, a seamless trial may be able to save time and reduce the number of patients needed for clinical development, thus reducing costs while also enabling investigators to learn about a product's efficacy and safety, and help regulators and sponsors detect efficacy and safety signals earlier in the development process. Phase 1 trials that are based on an enrichment design, in other words, trials where the patients are selected for treatment based on how their tumors express a specific biomarker, are associated with a significantly higher probability of demonstrating a clinical benefit than those that are not. We know that FDA guidance can be very helpful, facilitating product development and providing the clarity that industry and researchers say they desire. We recently issued for comment draft guidance on seamless trials and master protocols to expedite the development of cancer drugs. In addition, we issued a draft guidance on the use of placebos and blinding, which can pose ethical issues in cancer. FDA has generally advised that investigators use placebos only in selected circumstances. If possible, an active control is often preferred over placebo, such as an open-label trial with physician's choice of one of a few standard therapies, or a design comparing investigational drug to placebo with each added to the standard of care. All of these draft guidance documents specifically address cancer drug development. In addition, we recently issued 2 other draft guidance documents on clinical trial modernization, including the use of adaptive designs that addresses drug and biologic products more generally. Given the interest in novel, complex, innovative trial designs, including seamless trials, adaptive designs, and biomarker-enriched patient populations, we recently launched a pilot program that will give participating sponsors an opportunity to discuss these designs. Traditional eligibility criteria for participation in clinical trial sometimes excludes the patients most likely to be treated once the drug is on the market: the elderly, patients with poor performance status, organ dysfunction, brain metastasis, or other comorbidities. The percentage of patients who can access investigational agents is necessarily limited as a result of these practices, and enrollment accrual can be negatively impacted. This means investigators wind up chasing a small pool of potential patients, which can add to the cost and time of their clinical trials. FDA recognizes the problem, and in a recent draft guidance addressed eligibility issues surrounding the inclusion of adolescents in adult oncology trials. Some cancers in adolescents have the same histology and biologic behavior in those found in adults, but because of their age, adolescents are generally not eligible for enrollment, which delays their access to potentially effective therapies. A new approach may help, and we welcome comment on this recent guidance document. I know that Friends of Cancer Research has an interest in eligibility criteria as well. Together with ASCO, you recently recommended language for 5 guidance documents on ways to broaden eligibility criteria for cancer trials regarding minimum age requirements, trial enrollment, HIV/AIDS status, brain metastases, organ dysfunction, and prior and concurrent malignancies. FDA is currently reviewing these recommendations. Endpoints are another important facet of trial design. As you all know, surrogate endpoints are an important tool that can sometimes be used instead of direct measure of clinical benefit to support accelerated approval or traditional approval. As I mentioned already, in cancer, innovative endpoints such as progression-free survival and pathologic complete response are putting treatments into the hands of high-risk patients years before they would have been available using clinical endpoints, while still applying the FDA's rigorous standards for safety and efficacy. FDA just published a list of surrogate endpoints which were the basis of approval or licensure of a drug or biologic product under accelerated or traditional approval. It provides useful information for drug developers and endpoints that may be considered. Last month, we issued a draft guidance on the use of minimal residual disease as a biomarker in clinical trials of drugs and biologics to treat specific blood cancers. Minimal residual disease is a general measure of tumor burden. As a result of important workshops where we've heard from stakeholders and an analysis of marketing applications showing inconsistent quality of minimal residual disease data, the FDA identified a need to provide sponsors with guidance on the use of minimal residual disease as a biomarker in regulatory submissions. FDA is also at work with Friends of Cancer and other partners on the development of a biomarker of tumor mutational burden to evaluate whether it may help predict the likelihood that a patient with cancer will benefit from immuno-oncology therapies such as immune checkpoint inhibitors. Currently, there is a lack of standardization for such calculation and reporting. Different tests may report different measurements, and since there is currently no way of calculating tumor mutational burden, it is difficult to use as a biomarker. To achieve consistency and accurate reporting across tests, I know Friends of Cancer Research believes, and FDA agrees, that it is imperative to create some sort of standardization to arrive at clinically meaningful results, which will support informed decision-making for patients. Cancer therapies are typically evaluated based on their ability to shrink a tumor or extend life. But cancer patients have other meaningful concerns as well, such as coping with disease symptoms and treatment-related side effects that can impact their ability to function, or quite frankly, many other aspects of their quality of life. FDA's Oncology Center of Excellence has a patient-focused drug development program that is investigating ways to standardize and improve our measurement of these patient experiences, both during clinical trials and in real-world use following a drug's approval. Such information can be used to better describe the risks and benefits of cancer therapies and inform oncology drug development. For example, the FDA used information gleaned from patient-reported outcomes, or PRO, as evidence to support regulatory risk-based decisions regarding ruxolitinib, after learning that there were important differences in pain, itching, and night sweats in patients who took this drug to treat a rare disease of the bone marrow called myelofibrosis. FDA relied on another PRO measure to describe patient perception of the impact of ruxolitinib on certain visual side effects, such as floaters, shimmering lights, difficulty seeing at night. And FDA included this PRO information in the safety section of the FDA product label for this drug, which treats people with non-small cell lung cancer. Moving along the continuum of medical product development, let's consider FDA's advances at the early review stage. FDA is currently piloting what we call real-time oncology review. Before the applicant formally submits the complete application, they submit the final version of the clinical protocol and analysis plan, key dataset packages, top-line safety and efficacy tables, and figures and proposed labeling, giving FDA an early opportunity to address data quality and potential review issues. Along the real-time oncology review, we're piloting a multidisciplinary review template we call the assessment aid, which focuses the FDA review document on critical thinking and consistency and decreases some time spent on administrative tasks such as formatting. We're currently testing these two approaches with supplements before potentially expanding it to novel drugs and biologics. A cancer drug was approved using these two pilots in July. By the time the applicant submitted the application, the agency's review team had completed the analysis, and FDA was ready to approve it. Last year's approval of the two CAR T therapies for the treatment of advanced hematologic malignancies initiated what many hope to be a steady wave of new gene therapy products, including many cancer treatments. Indeed, we currently count approximately 700 active investigational new drug applications for such products. FDA responded in July to this fast-growing new field by issuing a suite of 6 draft guidance documents that we believe, once finalized, will provide a modern, comprehensive development framework for gene therapy. In contrast to traditional drug review, some of the more challenging questions relate to product manufacturing and quality, or questions about the durability of response, which often can't be fully answered in any reasonably sized pre-market trial. Issues addressed in these documents. For some gene therapy products, we may need to accept some level of uncertainty around these questions at the time of approval, with effective tools for reliable post-market follow-up. As we develop this evidence-based framework, we're going to have to modernize how we approach certain aspects of these products in order to make sure our approach is tailored to the unique challenges and opportunities created by these innovations. Nevertheless, FDA will remain steadfastly committed to regulatory paths that maintain the agency's gold standard for assuring safety and efficacy. In the age of precision cancer treatments, use of a specific test may help guide the use of a specific drug or biologic. FDA recently issued a draft guidance that outlines specific criteria under which sponsors may be able to include information about an investigational IVD and investigational new drug application submission to either our Center for Drugs or Center for Biologics. Allowing this pre-market information to be contained in a single application enables a more efficient review and further establishes the scientific relationship between the drug or biologic and the diagnostic used to select patients for treatment. Next generation sequencing offers another powerful diagnostic tool for cancer. Tests we approved last year can scan for hundreds of tumor gene variants to help match patients with therapies or promising clinical trials. We want to encourage development of these important tests by enabling a more efficient regulatory review. That's why we've opted for a policy approach that can keep pace with these fast-moving technologies, that gives patients and clinicians confidence in their analytical and clinical validity, while still allowing these sequencing systems to be efficiently updated as new genes or gene variants or improved algorithms become known. We've issued final guidance in April that explains how test developers may rely on clinical evidence from FDA-recognized public databases to support clinical claims while providing assurance of the accurate clinical evaluation of the test results. A second guidance provided recommendations on the design, development, and analytical validation of these tests. They are the latest examples of FDA's efforts to create regulatory efficiencies in the development and review of NGS tests. In 2017, we authorized a third-party option for conducting review of NGS tumor profiling tests and outlined standardized benchmarks for carrier screening tests to allow for their marketing without prior agency review. And important NGS products have already emerged. Last year, we approved tests that can scan for hundreds of gene tumor variants to help match patients with therapies and promising clinical trials. Earlier, I mentioned a guidance on the use of minimal residual disease as a biomarker. Less than 2 months ago, we permitted the marketing of an NGS test for minimal residual disease in patients with acute lymphoblastic leukemia or multiple myeloma. It measures the amount of cancer cells remaining in a person's bone marrow, either during or after treatment, and is useful in evaluating whether patients may be at risk of disease reoccurrence. I want to discuss one other aspect of our work across the continuum of medical product development: the FDA-approved product labeling, which provides essential information about the prescription drug. Friends of Cancer Research has developed proposals for the timely inclusion of new information on labels, particularly for generic drugs and older off-patent products, based on a study that showed that most cancer drug labels are currently out of date. We agree that updating labeling is very important. The fiscal year 2019 budget gave FDA funding to update the labeling of generic drugs, and FDA plans to begin with cancer drugs. I'm sure Dr. Janet Woodcock will have more on this when she speaks to you later today. Today I've taken you across a journey across the continuum of medical product development, discussing the many projects underway at FDA to build nothing less than a dynamic regulatory environment for those engaged in oncology research, development, and translation. Whether it is a biomarker development or clinical trial modernization, all of our efforts are driven by the same motivating goal: making the development process more modern, more scientifically rigorous, and more efficient, so that more safe and effective therapies can be delivered to the hundreds of thousands of cancer patients who are anxiously awaiting them. Before I take a few questions, if there's time, I want to make sure I keep you guys on schedule. One of the things I always offer when I have the opportunity to go out and share the incredible work that the agency is doing is to offer to also hear your feedback. We think it's very important to hear from those who are impacted by the policies that we put forward. So if there's also any feedback that you would like to bring forward, I am happy to bring that back to the agency. Thank you. Or you can save all your questions for Rick and Janet. I mean, that's another option too. Oh, it's coming around. such high attendance, you can't pass the mic around fast enough.
You guys shouldn't be shy, ask questions. So you're doing extraordinary work, retaining, attracting good people. How is that going? We had some fixes for that in 21st century cures, but that is extraordinary. And so that's part of the question. The other is that you're going so fast, so good, so smart. What about changing culture? How is that?
Okay, so the first question was around how we're doing on hiring and retaining the top-notch talent we need to be able to keep pace with the exciting innovations that we're seeing. And I will acknowledge this is an area of ongoing work. It's something that we are being very diligent in approaching. We've made changes internally to make sure that we're well positioned. It's a challenge. It's a challenge across the full breadth of our work. As you might imagine, there can be challenges and considerations that individuals think about when they're considering whether to stay in the private sector or commit to government service by coming to the agency. So it's something that you may have seen on some of the buses in DC. We even have a campaign out right now, FDA wants you, and it's kind of a superhero with a cape flapping in the wind. So, one thing I will say is that FDA is an incredibly special place to work, and you don't fully realize it until you have the privilege and the honor of being in the trenches and carrying out the work that we do on behalf of patients and consumers. And so I think that will always be an element that is distinguishable for the agency. And we're going to continue to do all that we can to make sure that we are well positioned to be recruiting and retaining the talent we need, not just now, but we're going to need it in the future so that we can keep up. Regarding kind of changing culture, I was chatting with Ryan and Jeff a little bit before we kicked off this morning, and I think that much of what we're seeing is also the culmination of a lot of work that has gone into this. I mean, this has been, if you were to ask the threshold question of, even going back to FADAMA, I'm going to take us back to 1997, right, because this is a recurring theme. One of the tremendously exciting aspects of science, medicine, technology is that we are just seeing an explosion of innovation, and with that innovation, an explosion of potential to harness these advances for the betterment of patients and consumers. And so it is always a challenge for a regulatory agency to be keeping up, and obviously recruiting and retention is part of that. Hiring and having the workforce you need to carry out the work is a big piece of that. But it's also making sure, as I touched upon in my remarks, that we have the right framework. And that also takes the opportunity of taking a hard look at, as these advances coming forward, how do we as a regulatory agency need to evolve, need to modernize, so that we are as well prepared and positioned to be able to play the important role that FDA will always play in assuring the safety and efficacy of the products coming forward for patients, and so that they can have confidence in the products made available to them. So I think of it also in terms of harnessing the potential. And I think we are seeing much of this. If you were to look at the work that Dr. Pastor is leading, I like to call OC the catalyst. I'm hoping this catches on at some point, because I think that they are a catalyst, in terms of harnessing from the medical product centers. And so as we see some of these ideas that we are piloting, we are testing, I think there's the potential to think about then how we could apply them more broadly, so that not just oncology benefits from these advances, but that other patients that face other conditions and challenges can also have the potential to benefit from them too. And now I'm 3 minutes over, but you guys will give me the sign when.
Hi, Anna. Great remarks. Thank you. I'm
Thank you.
Liz Mansfield, formerly of the FDA. And I wanted to ask you a little bit, you mentioned diagnostics in your talk about that there is now breakthrough status for medical devices. And I'd like to know how that breakthrough process is progressing. And in particular on the diagnostic side, whether there's any move towards making that center look perhaps a little bit more like Cedar in terms of having project managers and so on, because I know when I was there, people felt that they had a lot on their plate, and there's obviously a lot going on in the diagnostic sector.
Yeah, no, thank you for that question. So as you mentioned inherent in your question was that there was also a breakthrough program for devices, which I also had the privilege of working on the Hill. In fact, I think that was in part a testament to the very favorable response that the breakthrough therapy designation on the drug side had, so much so that this concept of applying this all hands on deck, when you saw very promising products on behalf of patients, then naturally carried over to the discussion with respect to medical products. And so it is a provision of the 21st Century Cures Act that our colleagues at CDRH have been very hard at work implementing. They were already working on the expanded access pathway concept, and so then with the statutory changes, they did conform in some of that. But I think it's, there are always going to be different considerations and characteristics and elements to drugs and versus biologics versus medical devices. It's kind of inherent in the very products. The innovation cycles are different. There are some cross-cutting, challenges that transcend all those areas, but then there are some that are unique. And I think one of the challenges that we face with respect to medical devices writ large is that their innovation cycles are different than the times that we see with respect to drugs and biologics. And so one of the things that Dr. Shuren has been hard at work with his team on is making sure that we really do have a modern framework that is prepared to appropriately calibrate the risks. And I think that that's also something that I think most physicians and patients would want FDA spending their time on the high-risk products. And so we're trying to think about how we can have, across the continuum of medical device products, how do we have in place the right architecture so that these promising innovations are able to come forward to patients and they're able to benefit from them. And I think one of the similarities that's happening in both the drug and the device side is how do we leverage in this modern era of medicine the potential for informing a product's life cycle. Jeff will talk about all the time, Dr. Jeff Shuren will talk about all the time, total product life cycle, right? And so inherent in that is how are you able to leverage what you can on pre-market for the considerations and questions that need to be answered? And then how can you think about then perhaps accommodating a little bit more uncertainty, a little bit more risk, because you're going to be able to capture information and meaningful data in your post-market setting. And then ultimately there's a loop cycle that comes back to continue to inform the consideration of medical products. And so that is another aspect of how we're looking at trying to strike that right balance in the device section. But obviously diagnostics are a pivotal part of patient care. And so we want to make sure that we strike the right balance there as well. I think Jeff is coming to give me the call. Yeah. But it really is a pleasure to be with you this morning. I hope you have a wonderful meeting, and it is just an honor to be back with you today. And I would just end by saying thank you for what you are doing on behalf of patients. I think every single one of us has been touched by cancer in some way, shape, or form, whether we know someone, whether we've been working to help advance policies to improve the quality of life and outcomes for those that do. And at the end of the day, it's all about patients. And so thank you for what each of you are doing on behalf of the patients you're serving. Thanks.